Okay, welcome to today's webinar, Spying on Insects with the Insect Eavesdropper. My name is Laura Isles. I'm one of the co-directors of the North Central Integrated Pest Management Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. We have a mission to coordinate IPM across the region by sharing and promoting all the cool things that are going on in IPM research, extension, and teaching. As we go through today's webinar, there's a Q&A portion on the bottom of your Zoom screen. So type in questions at any time, but it's just usually a bit easier. We'll go through questions after the webinar is over. So again, type in questions at any time and we'll go through those questions after the webinar. So I'm very excited to introduce Emily Bick from the Department of Entomology at University of Wisconsin-Madison, who's going to talk to us about her very interesting research. So I'll let you take it away, Emily.
Excellent. Thank you so much for the invitation to be here. I'm Emily Bick. I am the Assistant Professor of Precision Pest Ecology. I'm also the Extension Specialist for Field and Forage Crops in the state of Wisconsin. And because I'm new, I started a little bit over 3 years ago at this point. I like to start with kind of what the fundamental goal of our research is. We're trying to understand insect population dynamics. We're trying to basically understand how insects are moving and changing over space and time. We're then trying to translate that understanding into novel tools and strategies that enable, and you can pretty much fill in the blank here, but it's really data-driven agricultural decisions. And the approach that my lab uses is one that we've termed digital entomology. Now, a couple of years ago, this was fairly new, so people always ask, what is digital entomology? The way that I like to think about it is we try to incorporate sensors, we interpret the data streams of those sensors through machine learning and other kind of fancy, cutting edge statistical approaches. And because of my background in population modeling, we actually try to then help predict what's going on based off of both trapping and sampling data, as well as those sensors machine learning output streams. And the thing that kind of differentiates my lab a little bit is really how we interpret all of these data, right? We're looking at all of this through the lens of insect population dynamics. So, how insects are moving and changing over space and time. Now, I always have to start with, how do we get to this whole digital approach, right? I'm a classically trained entomologist. I've got a bachelor's from Cornell and my master's and PhD in entomology across the board from UC Davis. Uh, I basically started out working on mosquitoes. I actually joined a mosquito lab in high school, kind of in upstate New York area. I kind of think of those as like gateway insects. And as I progressed through college, through my schooling, I realized that I was really keen on the way that insects and people are interacting. I ended up working in the Midwest for a tree pesticide company for a 2.5 year period as a gap between kind of Cornell and UC Davis. I was working mostly on emerald ash borer. And in that process, I learned about how do we model insects? How do we actually predict? How do we help use these little computer simulations to make decisions? And I was a fairly computer competent. I took, computer science from high school and through college. So that was a pretty exciting approach for me to help landowners or decision makers or growers, add more data into that decision process. So when I moved out to UC Davis, I was working on in California, first in an invasive aquatic weed system, and then eventually for my PhD, I was working on Lygus hesperus in California strawberries, thinking about trap cropping. I was really trying to figure out how do we use all of this kind of known information, all of this sampling, all of these like sweet net locations, right, to make agricultural decisions. And that's where I found the big failing for insect models, right? Uh, Basically, the quality of data that goes into those models is exactly the quality of the usefulness of the output, right? Data in, data out. So bad information goes in, and you get models that are just not helping anyone. So from there, I turned around and said, my goodness, there's got to be a way to automate this. There's got to be a way to get better data streams. People, highly trained people like myself, can't be everywhere at all times. So that's when I moved out to Denmark. I was working on a Danish innovation fellowship, thinking about ways to automate insect monitoring. That brought me kind of all over the world, and then eventually back to UW-Madison. So the way I like to think about some of these insect sensors is there could be better data with sensors. Not better in all of the ways, right? Not necessarily the best insect ID algorithm. That one might not actually beat an expert. In fact, it probably won't at this stage. But we can certainly get better time and space resolution because, computers can be on all of the time. This is actually a picture of my lab group at not this past ESA National Entomology meeting, but the one before. But we were basically looking at all of these self-driving cars. I put my team over here actually in one of these Waymos so that we could, explore the city a little bit that way in Phoenix. But we're basically trying to figure out how can we use different types of sensors to monitor insects. And when I was a grad student, they were just starting to develop these types of vehicles. So when I was looking around trying to figure out how do we automate measuring insects, maybe measuring insects in flight, I found myself working with this group under the Danish Innovation Funding Scheme program, where we were thinking about how insects are kind of flying through different air volumes, right? So this is an entomological LiDAR device. It basically has an emitter and receiver. That red beam over there is essentially an infrared light. Most insects can't see red, so hopefully this doesn't interfere with any of their behavior. And it would, as insects would fly through that beam, the light would hit the insect and be reflected off on a single pixel photodiode. And you can tell what an insect was because when their wings are out, more light was coming in compared to when their wings were down. So then we would get this really, really incredible graph sampling that air volume 32,000 times a second, where we could get things like wing beat frequency or the relative body size to the relative wing size based off of those different elements. And get to things like species identification, in some cases, biodiversity metrics, matrices. And that was all well and good, except for this box cost about $30,000, and no farmer is going to monitor 18 liters of air volume for $30,000 at one spot in their field. So when I showed up at University of Wisconsin-Madison, I was really trying to think of kind of the cost-effective sensors here. How do we make how do we make really, really cheap insect sensors? So I was a little bit inspired. Um, I was a little bit inspired. I ended up in a sugarcane plantation in Indonesia within the first couple of months of my time in Denmark. Um, and basically, what we did was uh, I was challenged by these growers to do direct detection for insects. And at the same time, when I was at the sugarcane plantation, I was thinking really hard about how do we get around things like digging roots in corn or splitting stalks for European corn borer, digging roots for corn rootworm over there. So I thought about ways of doing this and ended up thinking about the Insect Eavesdropper, right? Like, why don't we just clip a very simple guitar mic? Those mics cost $1.25 to get. These days they might be a little bit higher with tariffs. We could plug them into sound cards, into aux adapters, plug them into these computers on a chip that's a Raspberry Pi, maybe add a screen and some power, and we could actually continuously monitor essentially the plant vibroscapes there. And what we have over here is actually our 3D printed case, right? So we have in the case, we've got an area where we can mount the Raspberry Pi, that computer on a chip. We can put in a USB hub, plug in sound cards, plug in those aux adapters, and then actually put out mics, drop in solar converter and memory. Those posts that you see over there are for wire control purposes so that the wires don't pull out. We can mount it on a stake in the field. And this is something that my grad student Alex Robus actually designed over here. And throw yeah, put a case over it, and run this continuously in the field for quite some time. This is actually something that we did over and over and over again. Last year, we actually had this out in 30 different sites. If you are an academic and you're interested in using this, well, we do have a patent on it. I also published how to build this and some preliminary software. We're putting together a much better software package end of June, but we published this with a free for academic use license because I want the sensor to actually be used in the environment. But we immediately are challenged by some pretty large issues, right? And the large issues start with what sounds are from the plant growing or the plant interacting with its environment or the plant interacting with other kind of biotic stressors, right? Other insects potentially, and what sounds are from our target insect. So what we did was we set this up with two separate plants. We had one plant that had a control and one plant that had an insect. And the basis of all the work that we've done has essentially compared a plant with no insects or as few insects as possible to a plant with insects. And immediately we ran into some issues. We're talking, what, 2023, February. We looked around the building. We could find tobacco hornworms, which are these large insects that will not stay in place. So we kind of had to cage them in specific areas. We could put this on Colorado potato beetles, which would try to escape. We tried this out with corn rootworm, right? But they were a little bit hard to get on roots without using eggs, and it's hard to quantify. So immediately I ran into some system issues. But pretty much across the board here, what we tried to do was show and actually determine what we could and could not detect, right? For these four insects, really early on, we were able to show that we could actually detect those large-bodied insects, both in Lepidoptera and Coleoptera, right? And what you're seeing over here is the amplitude, or essentially how loud is the insect, on two plants that are next to each other, run on the same insect eavesdropper device. The black is that amplitude for the plant that had insects, and the gray bar is the plant that control plant, the plant without insects. So immediately we could tell that we could detect at least these large insects. But as soon as we went to these smaller insects, things like European corn borer, we were looking at second instar that feeding on the leaves and third instar feeding physically within the plant. We could also tell something was going on. We could actually detect if the plant had the insect or not. The same thing with northern corn rootworm. Now, in many cases, we can actually try to match up the visual observations of what the insect is doing with something like what using camera data with the actual insect eavesdropper sensor. We've tried putting corn rootworm under a large microscope, and it hasn't worked out so well yet for us. We're hoping to redo that experiment. But in some cases, we can actually match these sounds together. And this is kind of the gold standard. But for the things that this insect eavesdropper is good for, right? Trying to determine what's a bite sound, what's a tap sound for a corn rootworm feeding underground on the root system, it's really, really hard for us to get that detection. However, we have teamed up with a number of folks, right? We've tried this out on 17 cropping systems, on 27 species of insects, on multiple life stages. We've partnered with 30 academic and government labs. We've contracts with two of the top five agrochemical companies. Some of the information of the folks we're working with is below. We've determined that we can actually eavesdrop outside, right? So what you see over here on the top graph in the center is a plant that had an in-furrow capture bifenthrin application, and below the control plant that was sitting right next to it in a corn rootworm field. We've also shown that we can hear piercing sucking insects. It's not just those chomping insects. So we've tried this out in aphids in the field, and then we replicated it again with Bob Cook's group in Minnesota and showed that we could definitively tell if soybean aphids are feeding on soybeans. But what's actually going on? Like what's going on under the hood? Well, at the base level, we get a waveform, which is this graph on the left, right? We have time on the x-axis and amplitude, or essentially how loud is the signal on the y-axis. we're running into some immediate issues because the microphones we're using are as cheap as humanly possible, right? You can get the parts for about 8 cents, and the entire unit with three mics costs about 120, so it's about $40 per plant that you're monitoring, right? So we're using the cheapest possible components that we can, which means the mics are not necessarily putting out the same amount of data. So for each microphone, we have to go and basically extract out the signals that we care about based off of what that baseline sound that the mic puts out is. And it differs from mic to mic. From there, we then turn this into a spectrogram, which is a 3D graph. And that 3D graph looks like what we see on the right. We tend to use that log frequency power spectrogram, where on the x-axis you have time, on the y-axis you actually have frequency, and then you get something in color of how loud or the power that that mic puts out. And once we normalize it, we can extract it out. We actually count the number of these extracted out events, and that's kind of our base number that we compare to. However, we'd still like to know what those sounds are. And we run into this immediate issue of what do we do about unground truthable data? Typically, when we're using kind of machine learning in ecology, machine learning in agriculture, we can use something called supervised learning, right? So I can tell that the top image of that bird is a peacock, the bottom one is a rooster, the bottom right, and kind of the bottom left is a duck, which means that I can then train an algorithm through my supervision, my knowledge, to split up those spaces, right? However, no expert in the world could tell me what a corn rootworm feeding underground on a plant sounds like, on a corn root sounds like. So we have to use some kind of novel methods. What we're doing is we're using these unsupervised learning algorithms to first add some structure to the data. So we take all the snippets, we run it through a highly pre-trained vision model because we're using these visuals now, those 3D graphs, those spectrograms, and add that unsupervised learning element. We then can start clustering them, where if sounds are visually similar, they'll actually start clustering together. But immediately we run into the same issue where we still don't know what each cluster means. So we take our segments, we run it through this unsupervised learning model, we cluster it, and then we're proposing a novel technique called implicit labeling. So how do we tell what those clusters are? And this is where our very rigorous experimental design comes in, right? We can actually take a look at the composition of the sounds, where each of the sounds in that cluster came from. Did they come from a plant without an insect, or did they come from a plant with an insect? Where, let's say once we've normalized all the sounds, they're kind of in equal measure. Uh, if 50% of the sounds are from a control plant, a plant without an insect, we know that cluster is not going to be from our target insect sound. However, and we have a strict threshold or cutoff of above 90%, if over 90% of the cluster's sounds are from a plant that had an insect, then we're in business. We're doing something right here. So we can kind of take those snippets, run it through a deep learning algorithm, cluster them, look at this implicit labeling, essentially using logic or inference to figure things out. We use our information about what's a bite, what's a tap, what's a noise, and all of a sudden we have these insect candidate clusters. I'm going to walk you through an example. The example I wish I could walk you through was corn one, but we had some technical difficulties. So I'm actually going to walk you through an apple example where Monique Rivera's group at Cornell basically took these apple bolts. They took a tree, they cut it into sections, they dipped it in ethylene, and they put on an insect and didn't put on an insect and left it on a lab bench to record. We're looking at, it was actually three different species of ambrosia beetles here. So first we go through that whole clustering algorithm and we come up with an image like the one on the left, where we know the different types of clusters and they're colored by cluster. And then we go and we label what the weighted percentage of the control is in each cluster, where if it's yellow, it has more than 10% control plant, and if it's purple, it has less. So all of a sudden, anything in red now on the right hand side is a potential insect candidate cluster. And anything in gray we can just throw out, we can ignore. We can then break each of these down and figure out, which insect was it, what was going on there. We can start breaking things down and getting some really detailed information about the time of day that these insects were active, and if there were different times of day based off of the different species of insects. And we can set this up in the field, which we've now replicated over 2 field seasons or in semi-field experiments, and double check and see if the field background or the sounds that we think are coming from the field line up with the lab background, or if the lab insects line up with the field insects. And it turns out across the 3 species, the insects were making characteristic sounds that this machine learning algorithm could actually separate out. Now, there were some sounds obviously in the field that just did not occur in the lab. We found some very interesting bird sounds, which we could hear through this. But this is kind of the approach where once we have the data, we can start to ask really interesting questions, right? When is the insect active? How is it responding to a treatment? Can we use this to scout without destructing a plant? Right? These are the types of questions that this type of tool, I'm hoping, will be used for in the future. Now, this work was not done in isolation whatsoever. Thank you to our funders. We've raised about 1.7 million in public grant funds on this project. Thank you to my lab. Dr. Mia Phillips has been a great postdoc helping push this project along. Lawrence Still was the former head of data science at the company I was working with when I was postdocing in Denmark, as well as to my grad students, especially Alex Arobas. James Kroll was really helpful. He's a UW-Madison professor. Monique Rivera, Brian McCormick's group, Rachel Arango, Sam Cook, Heather Grab, Dr. Cindy Cron, and Rodney Kruper. And I am sure I'm forgetting some, so I just put the et al. Signal down there. So I want to leave you with a question. I think there's a poll going around. If you could, what would you listen to? Any questions?
Thank you so much. And if anyone needs a continuing education units, that QR code should take you um, to that. So, and as we launch the poll, please fill that out. And if you have questions, this would be a good time to put it in that Q&A. I know it takes a few minutes to type into that Q&A box, so please do type that in. So I guess, I guess for my question, like, so it looks like you've kind of gotten to the like proof of concept, like this you feel fairly confident you can hear insects that you can't see on the plant, eating the plant.
Definitely some of them, right? So what we our next big paper push, which is going to be probably put together sometime January, February, is going through each of our collaborators' data sets and really answering the question, what is detectable and what is not detectable, right? So we know things like nematodes. We've tried this on 3 different nematode species in corn and in soybeans, and we can tell you without a doubt that this sensor is just not sensitive enough to detect nematode root feeding on corn and soybean plants. However, it would be really good to have kind of a systematic approach to kind of untangle these elements. One of the things that's been really interesting to think about is almost the plant structure as the limitation for whether this can or cannot be used, right? So things like soybeans are a more squishy plant, and this product or this approach is going to be less useful for some of those systems, right? We tried this out in a lab on soybean gall midge, and because of the way that the plant reacts to the soybean gall midge, the plant goes quiet, which means that we can't hear that boring insect. However, soybean aphids, which essentially don't hurt the integrity or the structure of the plant, we can hear pretty darn clearly. So it's going to be really interesting to kind of lay those elements out next to each other.
Excellent. So we have some questions coming in. So we have a question, would this device work on live trees without removing bolts?
That's a wonderful question. Yeah. So would this device work on live trees? So we have tested this out on a couple different systems with trees. We've tried it in apple trees in buckets, and let me see if I can go back over here to show you this initial picture. So we actually have tried it for two separate field seasons in these apple trees that are kind of nursery size. But we also had a small grant from the Maldives Space Research Organization, and we tried it out on coconut palms. We've tried it out on oil palms, and for boring insects, it seems to work really well, even though the tree is much larger than any of the elements that we're trying it on. So anything that's kind of woody or lignous or a little bit more dense, the sensor seems to work better on. Now, the big caveat for this is the larger the thing that we're measuring, the more outside sounds we would expect, right? So we tried this out on kind of large cherry trees and pear trees with Rodney Cooper's group in Washington, and it seemed to not work so well just because pear and cherry trees were just a little too large. They were touching each other, and it was really difficult for us to separate out that signal from the noise. Now, we didn't have this set of kind of implicit labeling approaches yet, so it's going to be good to go back and see if we can untangle that.
Hmm, very good. Next question. When this is deployed in the field, is the data able to be processed by the farmer on their own site, or does it need to be sent to your lab? I guess this is kind of getting to the
Yeah,
usefulness, or what are the
that
steps there?
that's a great question. So right now we have a prototype, right? This is a prototype that we mass deployed this past summer. I think we had about 75 of them out monitoring 30 different field sites across the US this past summer. So it's a prototype that works, and it works kind of long term for multiple months monitoring, assuming that you have good power. And I would not suggest the power brand that I have in this area. It just didn't work for us. Um, in terms of an actual product, I we are working on kind of licensing this technology to a spin out company. I'm sure other companies are kind of going to crop up doing very similar things. And once that happens, which is probably a couple years away, then it becomes useful for people like crop consultants or growers, right? That are curious about what's going on in their field and want to kind of do that non-destructive sampling. From the academic lab perspective, I'm basically going to run through proof of concept and best practices and what works and what doesn't work. However, the tool that I'm putting out is just never going to be useful on a production scale.
Okay, we have another good question. Interesting. How similar are the detection patterns for the same pest across different crop species? And they were thinking of diaprepes, root weevil. There's a root
Mm,
weevil that's on citrus versus sugarcane versus other vegetable crops.
how similar are the feeding patterns?
Yeah, how is
Well,
the sound similar on the plants?
yeah. So from a tapping versus crunching, it is pretty observable. So corn rootworm actually feeds underground on the plant, and it does more than feed. It actually uses its maxillary palps or its mouth parts to literally bang on the plant. In the 60s, they figured out that they had kind of a sugar sensing capability. In the 90s, they realized that you could actually use they could use their mouth parts to detect anti-feedants, which I would term as secondary metabolites or kind of plant compounds that stop insects from eating it, right? So they're using their feeding structures, their mouthpart structures as a way to sense. And those taps are actually highly distinct from something like a crunch. So the spikes that we saw in some of those earlier graphs are from taps rather than crunches. And you can tell because it has certain physics properties of when you like bang on a table, it kind of vibrates and dampens. So that's called a dampened isolation curve. And we could tell that between the taps or what we are hypothesizing are taps versus crunches. And we did all the testing on literally wooden, plastic, and metal sticks of different thicknesses and densities and all of that in the lab, literally tapping them versus, hole punching them, which was interesting. The other element is piercing sucking insects have a really distinct feeding pattern. So we've now tested what we think are Ligus linealaris, across multiple species, including cotton, which we have over here. This is exactly what the study is on, uh, but also in soybeans. And it has a really similar feeding pattern just looking at the waveform. Now, how does that hold up in terms of how is the sound actually being, um, I guess, deteriorated or affected by the plant structure? Like, that's the big question. And we're still at the really early stages of figuring out how, like, what is going on? Can we tell distance? Can we tell life stage? Can we tell species apart, which we could in the apple trees, but we're not sure we can do in other systems, right? We're still in the early stages. I was telling the folks that were on a little bit earlier that this past summer we collected 70 terabytes of audio data. And to put that into perspective, because that's too big of a number for an entomologist to think of, it was 17.4 years of audio data. So we are still unfortunately chugging through to try to figure out these answers. And I have a feeling we've got the data sets to work on for the next couple of years to try to untangle some of those relationships.
Really interesting. I was just thinking like we don't get to see the wood board. Like we can't observe. You're getting to observe things that we don't get to observe with the audio.
Definitely.
So another question, how will you decide what insects to add to your database? Will this be
Hmm.
decided by what crops are economically valuable to a given region, ease difficulty of traditional detection methods, like kind of, I guess, what are your methods?
Yeah, that's a great question. So we have a couple grants that are focused around the idea of building a sound library, right? So we've got one of those from the Wisconsin Soybean Marketing Board, building a soybean insect sound library. So for that one, we're literally building out the algorithms right now so that we can put them out in the world and say, at least on our soybeans in Wisconsin, at this particular plant crop stage, right, this is what they sound like, this is how you can identify them. Now, we're not sure how broadly applicable those algorithms are, which is why we're being really careful in putting them out for academic use, but not for commercial use at this point. I think whatever company ends up running with this idea, since it seems like a popular enough idea to be commercialized, um, they will probably put out some sort of universal, insect ID classifier, and then from that, a rain classifier, and bird sound classifiers, and then start to go into subgroups like, is it piercing sucking? Is it taps? Is it crunches? Right? Telling how far apart these sounds actually are. But I think from that perspective, I'm working with all of kind of the best scientists that I could find that could play with my equipment for under somewhat limited resources. But and my region is going to be focused more on corn and soybeans, just because that's what I'm supposed to be focusing on as part of my extension group. But it is up to the company that's commercializing this to figure out what the right path towards what the right crops are appropriate. And I'm hoping that the idea takes off because it seems like a good one.
Mhm. Um, Thank you. Our next question is, could this be used to detect structural pests, termites, ants,
Mm.
etc.? How about bed bugs?
Oh, okay. So almost certainly not bed bugs, I think is safe to say. We have tested this out on termites. It works. It seems to work very well from our preliminary data on termites, right? We've been working with, there's a U.S. Forest Service, well, she's got an adjunct professorship in our group. This is Rachel Arango over here. She's U.S. Forest Service. She sits in Madison, Wisconsin, and been doing experiment after experiment after experiment on termites. And it seems like for woody structures, it's a pretty clear indicator that we can actually tell what's going on. However, we're just getting the giant database together, and we might be completely off base here. And we have no idea if that will actually translate to like a house size or a structure size or a specific area, or if this is going to be an appropriate tool for that. However, something like bed bugs that are not actually actively feeding on a structure, and it seems to be unlikely that we'd be able to detect them.
Picturing you put little sensors on the people and see if you can. There'll probably be a lot of background sound. So
We did try
again,
this with stethoscopes early on, and it turns out they work a lot worse than the contact microphones.
again, if there's any questions, again, please type it into that Q and A portion. Is So the next question is the camera component required for functionality, or can this work exclusively on audio data collection?
The camera that we have out there sometimes is entirely for validation. It is unnecessary for the sensor.
Any other question? I mean, like kind of are there thresholds? Like, is there kind of like a
Mm.
minimum number, or like, do you think in the future you'll be able to tell like, okay, there's 10 aphids versus 10,000 aphids on this plant?
That's a great question. That's actually what our postdoc Mia Phillips is working on, that density question. We've got a grant between my lab and Brian McCormick at K-State's lab, literally trying to figure out, um, yeah, can we tell the difference between density? And the answer seems to be maybe, right? So insect behavior is not necessarily insects have density dependent feeding behavior. So the current hypothesis is the 0 to 1 is going to be really clear, the low to high is going to be really clear, and that it will actually asymptote out where there's going to be above a certain number where the insects are regulating their fellow insects' feeding behaviors, right? Because at that point, at a certain density, they're actually going to want to move and they're not going to want to eat from that plant. So, whether we'll be able to go through a traditional threshold or we'll have to come up with a feeding rate to try to use this to make decisions is it's still a little bit up in the air, but we're a year into that grant.
Very good. And have you tried this on like Emerald Ash Borer or anything?
Oh, you're going back to one of my
Your
first major
first,
pests that I
yes,
worked on. I think I might have 7 publications on Emerald Dashboard. We have not tried this on urban trees, in large part because that's not what my focus is. Crops are, yeah, of higher import to me right now.
fully understand.
Uh,
Any other questions? I'll give you all a few more minutes to type in any last questions, but thank you so much. This has been really interesting, and we will get those poll results to you. And I just want to remind anyone that our next Pest in Progress webinar will not be until January, so not until 2026, so January 21st, get at 12 p.m. Central, 1 p.m. Eastern, and we're going to discuss the breeding effects, breeding efforts to curb cucurbit downy mildew, a little bit of a tongue twister with Edmund Frost. So see everyone in 2026. And I don't see any other questions. So thank you so much, Emily. We really appreciate this. It's been very helpful.
Thank you all.